Hi, so now this is the second half of the metabolic test for identification of bacteria. We'll finish up the gram-positive cocci tests. That's the mannitol salt agar. And then we will begin the gram-negative bacilli tests. We're only doing 2 of those, and the first one is the coliform analysis. There we go. So metabolic tests for gram-positive cocci include the ones that we just did, catalase, and coagulase. And the 3 gram-positive cocci, once again, that we're looking at are Staph aureus, Staph epidermidis, and Enterococcus faecalis. All 3 of which are gram-positive staphylococci, can't be differentiated on a gram stain, and for that reason we do the metabolic test. So last time we said that the catalase test, the Staph aureus had really vigorous bubbles. It was positive for catalase. Staph epidermidis, also bubbles, not quite as vigorous. And so it too is positive for catalase, but that Enterococcus faecalis, no bubbles, no catalase. Okay, so no bubbles means that the hydrogen peroxide was not converted into water and oxygen. So remind yourself what the equation for that is and why the word catalase is written over the arrow. So we said that Enterococcus faecalis is an aerotolerant anaerobe. It is negative for catalase, and so that's one of the ones that we could eliminate, at least with the catalase test. But with Staph aureus and Staph epidermidis, you just don't know if they are obligate aerobes or facultative anaerobes, both of which are positive for catalase. We said last time in the other PowerPoint that with obligate anaerobes and microaerophiles, their problem is they can't grow on the surface of a plate. So the very fact that we are working with colonies growing on the surface of a nutrient agar plate means that they, just by being able to live on the surface of a plate, they can't possibly be an obligate anaerobe, nor could they be a microaerophile for whom normal amounts of oxygen are lethal. Okay, so we then have figured out that at least Enterococcus faecalis is an obligate anaerobe, but we can't tell the difference between Staph aureus and Staph epidermidis on catalase. We did see that there's a difference between them if we do coagulase, but what if you didn't have coagulase? It's not that easy to get plasma. In fact, that's one of our most expensive labs that we do, is getting that rabbit plasma. There is another test that you can use to tell the difference between Staph aureus and Staph epidermidis. Remember that both obligate aerobes and facultative anaerobes have both catalase and superoxide dismutase, so catalase is not a good way to tell the difference between them. When we look at facultative anaerobes, remember we showed this picture in lecture, and they do prefer an oxygenated environment. That is the way they prefer it. They make a lot more ATP. That's the reason for that preference. But if there isn't any oxygen available, remember that the obligate aerobe would grow as a thick amount where oxygen concentrations are highest. So here at the top of the tube is where you would be seeing that. Excuse me, let me see if this will help if I do it over here. The obligate aerobe has the most amount of colonies up at the top, nice thick layer, but then after that you don't really see anything because they would die. This is what would happen to any of us if we were put further down the tube where there isn't any oxygen to be had. When you're talking about facultative anaerobes, both these 2 tubes in the center, tubes 2 and 3 in this case, going from the left to the right, are both facultative anaerobes. But you can see that being a facultative anaerobe does not mean you have equal abilities in low oxygen conditions. We can see that of the 2 tubes in the center of this picture, the one that is on the right, which I would call tube 3, that tube, obviously you see growth all the way to the bottom. So even at the lowest oxygen concentrations, this facultative anaerobe has a greater ability to grow with lower oxygen amounts than the tube just to its left, which is also a facultative anaerobe. So being a facultative anaerobe does not mean you have equal abilities. Okay, so if I can give you a terrible analogy, you know, I'm a runner, so is Usain Bolt. Like, we're not, okay, right? Yes, I could say I like to run, and so does he, and we obviously have really different abilities. And I know you've never seen me run, but it's not that best. All right, so facultative anaerobes. All right, so in lab, or excuse me, in lecture, we said that the one on the left, this is Staph aureus, and the one on the right is E. coli. So it kind of giving something away right there, but anyway. And then over all the way to the right, the obligate anaerobe over here, you see it's just almost crystal clear right through the topmost part of this broth tube, and it's because they can't deal with oxygen at all on any amount. It's growing really well below. In fact, I could actually see big clumps down here. Remember, if you can see it, that represents a ton. But all of this is all frothy, and you know, I can see stuff in there. And so wherever there isn't oxygen, they're quite happy. Okay, now facultative anaerobes, what they will do up at the top is they will do a normal cellular respiration. They will use oxygen as the final electron acceptor. They will make 38 ATPs. They love it there. That's their happy place. Without oxygen, you can't run that system. We said what the problem would be, that you just could not pull on the electrons. You cannot make a hydrogen ion gradient. You cannot make ATP. That's death for the obligate aerobe, but for the facultative anaerobe, that's okay. They'll just go ahead and ferment. Fermentation produces acids. We looked at that table before. I asked you to know one organism and its final product from fermentation. And for many of you, you chose some acid or other. Some of you chose other things, but nevertheless, fermentation will produce acids. That's one of the things that we know. Okay, so with mannitol salt agar, this media is selective and differential. So remember that when you're talking about chemically defined media, there are three kinds. Chemically defined means I know exactly what is in there. In fact, in the mannitol salt agar lab, there is a table that gives you the actual concentrations of the components of this agar. So mannitol salt agar is selective. That means it enhances the growth of the target while suppressing the growth of the unwanted. And then there's differential media, which would give a visual distinction. So mannitol salt agar is both selective and differential. Mannitol is a sugar. So you've heard of like sorbitol and things like this. So sometimes some sugars are named in that way, rather than ose, like glucose or fructose. So here's mannitol. And so it's a sugar, and that is a fermentable substrate. So if there is fermentation, it'll show up as an, because we will be able to test for acids. There's also salt in here, as mannitol salt lets you know, and the salt is 7.5% salt. So the ocean, you know, you've been there and you've probably been in, and maybe you got some salt water in your mouth and it's so salty, you just, you know, it's terrible, whatever. The ocean is 3.5%. This is 7.5%. 7.5% salt is really, really salty. Remember what salt, high salt or high sugar, would do to an organism. Remember that that is too much osmotic pressure, that landing on a highly salted fish or highly sugary honey would mean that that bacterium would yield its water because the external environment is hypertonic compared to the cell itself, and so the cell would yield water and die. So putting 7.5% salt in there is really selective. Only halotrophs or halophiles will be able to live. Everybody else is going to die. That's the definition of selective, isn't it? That it will enhance the growth of your target and suppress the unwanted. So always with selective media, you must say, how is it selective? How is that happening? So the selective pressure here would be the salt, the 7.5% salt. Should you know the percentage? You absolutely must, because it is that high. And recognize that the ocean is 3.5. This is more than twice as salty. So that's a lot. All right, so if I'm going to test for fermentation, I'm going to be looking for acids, which means I need to have a pH indicator, and that's my phenol red. I really like phenol red because the two first letters pH indicate that's what it's doing, it's a pH indicator. So I'll be able to tell, it'll be a color change, and the phenol red will let me see that. And then lastly, in mannitol salt, there's peptone. Peptone makes you think of peptide because peptones are really just short little pieces of proteins. It is a source of amino acids. Okay, so what is there to eat in here? Well, the agar is indigestible. That's been established for us before, that is a polysaccharide composed or made by red algae. And yeah, polysaccharide would be a bunch of glucoses together, absolutely, but they're linked together in a way that bacteria cannot break down. That's why it's used as the solid substrate in laboratories. But, so, okay, the agar is not edible. What else is there to eat? There's sugar and there's peptone, and that's it. There's no lipids, there's no other sugar, there's no nucleic acids, there's not a bunch of yeast and extract and beef extract. This is it. So this is chemically defined, really specific. Okay. Okay. So if I'm checking for fermentation, then what I need is a test for the acids. And it turns out that phenol red, my pH indicator, will go yellow if the pH drops below 6.8. So 6.8 or below, it will be yellow. And so here in the image, you can see that one of the quadrants, so if it was split up into fours, that this area is yellow. You can see growth on here and you see yellow all around. This is an indication that these bacteria growing here in this quadrant are producing acids, and those acids make the phenol red go from kind of a pinky orange is how it starts out into a yellow. That's visual. I can see that from across the room. You could hold up your plate. I could say, I see yellow. That's the visual. That makes it differential. Differential because of the phenol red. So if I see acids, that will tell me that the mannitol has been fermented. Fermentation produces acids. What about this color just to the right in the top right quadrant? Okay, so if we're looking at, if you remember your math of this is quadrant 1, and the yellow would actually be in quadrant 2, wouldn't it? Okay, so quadrant 1 is really pink, like really pink. It's a deep pink. It's like a fuchsia or a raspberry. That happens when phenol red is in the presence of bases. If it's over 8.4, so you remember 7 is neutral. So if it's over 8.4, that's very basic. The phenol red will turn a very pink color, fuchsia or raspberry. I'm sorry we're not seeing it. It's really pretty, actually. So I can see that that's happened, okay, if I see the pink. Over here in what would be quadrant 4, so just below the fuchsia, it's the original color. It's blank, kind of nothing. Okay, so for this test, then, I have a way to check for fermentation. I have a fermentable substrate is the mannitol. It's a sugar, which could be fermented. If it is fermented, I'll see it because of a color change to yellow by the phenol red. And if they don't ferment, they'll eat the acids in just a normal way, cellular respiration, which produces only water and and carbon dioxide. Neither one of those products is acidic. And once you're out of sugars, what else is there to eat? Well, that would be the peptone. And peptone, the breakdown of that will give us an amine that is going to be something that is basic. And so I would see fuchsia or raspberry. Okay, So mannitol salt agar has 7.5% salt in there. That's just too much osmotic pressure for most organisms. That is a lot. That is a lot. So most organisms cannot grow on this plate because it's just too salty. Only the halotrophs or halophiles can grow here, okay, grow easily. This is what makes mannitol salt selective. Okay, so what's the procedure? Well, it's essentially very similar to what we did with the catalase test, okay? Now, in this case, we're doing the Enterococcus faecalis on this test because it just acts as sort of a negative control. But it would be the same thing. You would flame your loop, okay, and then flame the tube and insert the loop and take out a sample and then make a little zigzag pattern in each third of this plate. One for Staph aureus, re-sterilize the loop, do it again for Staph epidermidis, re-sterilize the loop, do it again for Enterococcus faecalis, and incubate overnight at 37. When we would come back, this is what we would see. So I couldn't find a picture that only had 3 quadrants. This had 4, and that's actually probably better because here's the fourth in this case is no streak. So you could just see the color of the mannitol salt agar initially and see whether there's, what you could compare to the rest of them. Okay, so what do we see? Staph aureus, I see very good growth, so it's not deterred by the salt. Okay, and we know this. It was our exception, our outlier, right? Exception, excuse me, when we were talking about how osmotic pressure could stop most organisms. That doesn't work on Staphylococcus. It grows just fine. But more than just growing, I see yellow. Yellow is my evidence that this area is acidic because phenol red will turn yellow at pH below 6.8. So this is acidic, absolutely. And so it has fermented the mannitol. And so this is something I can see very easily. Staph epidermidis, just over here to the right, which would actually be quadrant 1, is very, very pink, fuchsia or raspberry. I see plenty of growth. I can see the organisms just fine. And all around this whole quadrant, it is fuchsia or pink, indicating bases. So remember that when you break down, so I don't see any evidence of fermentation, do I? Fermentation would be yellow. I don't see yellow here. I see pink. So can Staph epidermidis ferment mannitol? The answer is no. If it could, then this would be yellow. What it did do is eat the sugar first. All organisms eat sugar first. This is our problem, isn't it? It's like your carbs are going to be the first thing broken down, and then it would be lipids. But there's no lipids in this plate. And then you go and break down the proteins. Okay, so after it eats the sugars aerobically, remember that the end of aerobic respiration gives us oxygen and carbon dioxide as the waste products. Neither one of those things, it gives us water and carbon dioxide as the waste products, and neither one of those are acidic. Okay, it would eat the sugar, and what else is there to eat? Peptone. Well, the peptone, every time you break down the amino acids, then the amine group, the NH3 group, that is a waste. So if you remember that slide from the metabolic abilities of bacteria, and we said that, you don't just eat sugars, you also eat fats, you eat proteins, and what happens to them and where do they join in glycolysis and Krebs and, etc. We showed that the amine group was a waste product, and I made a point to indicate that. That amine group is basic. That's what turns this phenol red fuchsia, is the breakdown of peptone releasing the amine. This is my proof positive the Staph epidermidis is already eating peptones instead. Okay, who else is on this plate? Enterococcus faecalis, and what you can see is that you can barely see any growth at all. This is having a really hard time growing here, and yes, it's got a problem being in oxygenated environments. We already established that before with the catalase, but the other problem that is keeping it from growing is the salt. It's just too salty here. So Enterococcus faecalis is having a really hard time growing on the surface of a mannitol salt agar plate. much less showing any ability of metabolism whatsoever. There's almost no change in color between it and the blank, as though nothing were put there at all, because it's having a hard time living, which means you're not seeing any evidence of metabolism whatsoever, either fermentation or the production of a base. You're seeing nothing because it's having a hard time living. Okay, so move forward. On page 96 in your book, you would see that these tests, metabolic tests for gram-positive cocci, would be laid out in this format, which is known as a dichotomous key. A dichotomous key is a really fancy way to just say, like a flow chart. So that if you would start with a gram-positive cocci, now on page 96 it says you take an organism, you do a gram stain, and it would be either gram-positive, it would go to the left, and gram-negative, it would go to the right. Okay, and between the gram-positives, are they cocci or bacilli, and we'd be following the cocci. First, you do a catalase test. If you're negative for catalase, you're Enterococcus faecalis. If you're positive for catalase, well, and we saw that here, you could be either Staph aureus or Staph epidermidis. I could tell those apart in two ways, the coagulase test and the mannitol salt, both of which the Staph aureus is positive for fermentation of mannitol and positive for coagulase. I would get a clot in the plasma. Staph epidermidis is negative for both the fermentation of mannitol, and it is negative for coagulase. It does not form clots. So this is how you can tell the two of these apart. Now, one thing to note here, the fact that Staph epidermidis cannot ferment mannitol does not let me say, oh, it's an obligate aerobe only. There's a ton of fermentable substrates. I cannot make that determination from mannitol alone. Just because it can't ferment mannitol doesn't mean it can't ferment glucose, or that it can't ferment fructose, or any of the other sugars that you could come up with. The answer may well be it can't ferment anything, but I can't determine that from mannitol salt agar. So this is the really unfortunate thing of us not being able to meet and see each other, because this is the kind of thing I hammer home in these tests. Every test will let you make one, maybe two conclusions. You can't leap to all kinds of other thoughts. So not being able to ferment mannitol does not mean it can't ferment anything. I cannot make that determination. So we are very careful on what we will agree to say yes or no to. Frankly, in the end, knowing whether or not Staph epidermidis ferments or can't ferment, that's not a big deal. What this is training for you, because eventually you're going to be looking at tests that were done on blood, tests that were done on sputum, tests that were done on stool, or what have you. And you've got to be able to say, I can conclude this. And people will try to press you. Does that mean this and this and this? And you're saying, you know what? I can't conclude that. The test only allows this conclusion. What tests often lead us to is more tests. So this is where we are, and I just really want to make sure that we understand that just because it's yes or no to a specific test, you can't make huge conclusions, okay? Now, let's just think about this for just a minute before we go further and move away from gram-positive cocci altogether. And that is, if you think about where these organisms would naturally be found in nature, then the results of the tests that we have done become very obvious. Staph aureus and Staph epidermidis can be found, guess where? Yep, on your skin. Aureus can also be found in the nasal passages. The skin and the nose are highly oxygenated environments. Organisms that live in highly oxygenated environments have got to have mechanisms of dealing with reactive oxygen species, so I'm not really surprised that they are both positive for catalase. Enterococcus faecalis. So the first part of this last name here, the second name, makes you think of feces because yes, you find it in the colon. The colon is not particularly oxygenated. And so this guy, Enterococcus faecalis, doesn't have catalase. It's kind of a waste for it to have it. It wouldn't really encounter reactive oxygen species in the lower intestine very often. That's just not a very oxygenated environment. And so it lacks catalase. Not only that, okay, so that helps us understand the reasoning for the catalase, yes or no, with these three organisms. When you're looking at the ability to grow on the surface of mannitol salt agar, salt, 7.5% salt, where do I have salt in my body? On my skin. And some people's skin is, way saltier than others. You've seen their crusty, baseball caps in the summer, etc. Staph epidermidis and Staph aureus live on the skin. Our sweat is salty. They're okay with that. You know what? That salty sweat, that's actually a good thing because that's keeping most organisms away. They can't handle living on the surface of your skin because it's too salty. So look at how you're built so perfectly. Isn't that wonderful? That all sorts of stuff could be landing on you and, good luck. You're really salty. Are you 7.5% salty? Maybe not overall, but what about right at that sweat gland? What about right there? It's probably pretty high. Okay, so there we go. That's our answer for why these guys can live on salt. And Enterococcus faecalis, it's not salty in the lower intestine. No, it has a really, really hard time with that. So this is the end of the gram-positive cocci metabolic tests. And remember, why do I have to do these anyway? Because I can't tell them apart because they're all gram-positive staphylococci. Taking a gram stain, well, that's nice. I mean, okay, they're not gram-negatives, that's nice. But I can't tell the difference between these gram-positive cocci by looking. I have to look inside. And for that, we do these metabolic tests. Okay, so moving on. Go now to the identification of gram-negative bacilli. And the first one of those is the coliform analysis. So the coliform analysis is a three-parter, and they each have names: presumptive, confirmed, and completed tests. We have to know the names, I'm sorry to tell you. So the presumptive, the confirmed, and the completed tests. So there's three tests for the coliform bacteria, and coliform, that also has a definition which we do need to know. It means having E. coli-like characteristics, and that means that you're just like E. coli. If you're a coliform, you are gram-negative, you are bacilli, you don't make spores, so you're non-spore forming, and you ferment lactose with acid and gas production. Okay, so E. coli is prevalent in the lower intestine of humans. It's about 1% of the bacteria found in your feces. And let me tell you, there's a ton of bacteria found in your feces. E. coli is only 1% of it. So what's the other 99%? Well, that depends. What do you eat? What do I eat? Where do you live? Where do I live? Do you have pets? What are my pets? Okay, what meds are you on? What meds am I on? How old are you? How old am I? Okay, all of these things influence the composition of what your normal microbiota might be. So I can't agree with you on the other 99% of bacteria found in feces, but we all have E. coli. And so because of that, E. coli becomes the test organism for feces. E. coli is typically not harmful. It's actually one of your friends in your lower intestine, okay? Normal microbiota. But because it is found in human feces, commonly found, everybody's got it, I can test for the presence of E. coli, and that would tell me that feces is present. So we test water samples for E. coli in the coliform analysis to see if that water sample has feces in it. I am not concerned necessarily with the E. coli. In most cases, E. coli is no problem whatsoever. Of course, there is the E. coli 0157. Every once in a while, you hear about it. I think the last time, I think it was in spinach. Sometimes it's romaine lettuce. It was melons not that long ago, probably 6-8 years ago at this point. There is a bad form of E. coli, but typically I'm testing for E. coli, not because it's the harmful one. What I'm worried about is if there's E. coli in the water, that means there's feces in the water. And now I'm worried, like, what else is in the water? Because we're going to find out in our pathology, pathogenic bacteria lectures, that fecal oral contaminants are plentiful. There's a lot of them. So in this lab exam, I will ask you to name 4 fecal oral diseases. I will name 2 for you right now, and those 2 cannot be used. One of them is cholera. You know about this already from the video that we watched independently. We watched them at home. That cholera, and remember they cleaned up the water source, and by doing that they got rid of cholera. Cholera is fecal oral contamination. Another one is polio. Polio, teeny tiny virus, naked RNA, right? Really, really small virus. That is fecal oral. It's super hard to find virus. You know that. It's so small. And then I can't like isolate it and grow it that easily. I have to put it in a tissue or in eggs. This is a big problem. I can't find that very easily. But what I can find easily is E. coli. E. coli would tell me there's feces in this water, and then that water becomes undrinkable or unavailable for recreation. No swimming in this beach, no swimming in this pond, etc., because of what else might be in there. Okay, so it is a 3-part test. The first one is known as the presumptive test. And the presumptive test, see what that means? Presumptive. I already assume, I presume it's in here. Guys, let's just like assume that this water sample is contaminated. So I would have asked you to bring in a water sample, and the best water samples to bring in are like well water. In fact, the coliform test that we do is the exact same one that the county does on your well, on your property if you have it. And so typically people would bring in well water from their house, and from the tap in the house, and well water from the well itself. And that's kind of a good test because then you could see if the filtration is working. You can also bring in pool water, like a public pool. You can also bring water in from a beach or a pond or a river or a creek or whatever. Those were the samples that I would ask you to bring in. And okay, so we would test this water sample, and we would do it in 3 different tubes. Okay, the tubes would have lactose broth. That would be the only thing in here. It'd be lactose and water. Okay, lactose is a sugar, and it's a food source. Okay, lactose broth, it is the sole carbon source. That's the only thing to eat in other words. Okay, all organisms are carbon-based. All 4 carbon compounds have carbon in them, don't they? And so they need carbon and carbon sources. And in this case, this is all there is to eat. So if that's the stringency, then this test is already selective, isn't it? If you cannot metabolize lactose, you cannot live in here. So it's selective, and this is why lactose is the sole carbon source. There's nothing else. There's no protein in here. There's no lipid. There's no nucleic acid. This is it. Can you survive on lactose alone? That's the question right here. And so we would take your water sample and we would put 10 milliliters into the first, then 1 and 0.1. And this is kind of a way to do a little bit of a qualitative and quantitative analysis of your water sample. Remember to be a coliform, not only would you ferment this lactose, but you would also produce acids and gas. So acids, I am not going to be able to see. I don't have phenol red in here or anything. There's no acid test, but I can check for gas with something called a Durham tube. A Durham tube is a small glass tube that has the opening at the bottom and the closed part goes on top. So you sort of put it in there upside down in all three tubes. You would add the 10 milliliters of your sample to the first tube, 1 milliliter to the second tube, and 0.1 to the third tube. And we would just incubate it overnight and see. So what kind of results would you get? Well, so we can see that in these tubes, all of which are lactose broth, okay, so this is the presumptive test. Notice that in this case, this is the way I put it together anyway, that I would have some turbidity. It's not clear in this tube, indicating growth. But more than that, I have gas in the Durham tube. I have it in the 10 milliliter tube, the 1 milliliter tube, but the 0.1, I don't see any. So remember, I'm looking for the presence of E. coli. So it looks like it might be in these two tubes that both have greater concentrations of your original sample than the last tube, which has a tiny amount. So if you do have a fecal contaminant going on in your water sample, your well, your pool, whatever, it might not actually be that bad, because if it were a really bad situation, it would be present even in the lowest concentration. So I hope you understand that. So in the presumptive test, the only way to pass is with gas. Gas is the pass. So this last tube, the 0.1, that's a fail. There's no gas there. That's a fail. Okay, so for some people, if we had done this in lab, some people, you come in with your well water, gee, I hope everything is fine. And, you know, all three tubes would have nothing, no gas in any of them. And you could leave and say with certainty that there's no E. coli and therefore no feces in that sample. Okay, those people would be happiest of all. This is one of these tests you want to fail. Other people come in and they do this and, oh, I have gas. Oh, I have gas. I think I may have feces in my water. I can't know for sure yet, though. This is a 1 of 3-part test, okay? We would go forward only with tubes that have gas in them. So if you were somebody whose all three tubes showed up and you were negative, you'd have to borrow somebody's to go forward, okay? To go forward, we would take a sterile loop full of the sample that has gas, but the least amount of samples. So which one would I choose here? You have to have gas, but the least amount of samples. So yes, it would be the 1 ml sample. In this case, we would go forward with one sterile loop. So then we would go on to the confirmed test. So it takes like a while to get through the coliform analysis because it's 3 tests. So the confirmed test would take this positive sample that had gas but the least amount of water sample in it. In our case, it was the 1 ml. You would take a sterile loop and you would put it into the brilliant green lactose bile broth. Boy, what a mouthful. Brilliant green, that's the dyes in there. Lactose, still the sole carbon source, and there's bile in here. Okay, brilliant green lactose bile broth. It is a beautiful color. We are really missing out because, and I should stop saying that, I'm sorry. We're just making the best of what we have, but it's like this emerald green. It's so pretty. Anyway, it also has a Durham tube in here, this upended glass tube with the opening at the bottom and the closure at the top, so sort of upside down. You would take a sterile loop full of this and put it in here. The brilliant green dye and the bile both inhibit gram-positives because in this first test, okay, with your lactose, you're looking for a lactose fermenter with gas production. And what I can see with gas is I have fermentation that has resulted in gas production. But I don't know if it's on gram negative. And to be coliform, you must be gram negative. So I'm going to inhibit gram positives with the brilliant green and the bile. Both of these, the dye and the bile, inhibit the formation of peptidoglycan. Peptidoglycan, you remember, is the cell wall, and gram positives have a very thick cell wall, and that is their outermost layer. If they cannot make a good solid cell wall, they will die. So they will not survive in brilliant green lactose bile broth. So we put 1 loop full of this lactose broth culture with the gas. Okay, your water sample that gave you the gas. Put 1 loop full into brilliant green lactose bile broth and incubate 37 degrees overnight. And then it would be coliforms part 3 and 4 would be the following week. We can only go forward with gas. Gas is still the pass. Okay, so if we look at these tubes here, what do we see? Which one has gas? Only tube number 1. So this, you would only have gone forward and done 1 tube. I'm just saying if you looked at 3 people's tubes together, say. Only gram negatives can survive here. So this tube here, I don't see any turbidity or change or whatever. So whatever was in, now remember, the only reason I could have gone forward is if I had gas. So if I had gas and went forward into the brilliant green lactose bile broth, but now I have no gas, then I know that whatever gave me the gas before must have been a gram positive, and now I just got rid of it. With the bile and the brilliant green, I inhibited the ability of that gram positive to make a cell wall, and so now they're dead. They did not survive in here at all. Okay, I couldn't have gone forward if I didn't have gas. All right, what about this guy? Well, this person went forward with gas, and now I see turbidity, but I don't see any gas anymore. This is a fail. So there is definitely growth in here. So that means there's gram negatives doing just fine in here, but whatever gave me the gas before, that must have been the gram positive because now it's dead. And now I don't see any gas anymore. Both of these are over. So the person whose well water came up positive in the presumptive test in the lactose broth, very concerned, oh my gosh, there's feces in my water. And I would say, hey, you don't know yet. Here, at the end of this confirmed test, they would go home happy knowing that whatever is giving them gas in the lactose broth was a gram positive, which means this is not fecal. It's not E. coli. It's not fecal. And so what you would know is you have bacteria in your water, and you probably do. Okay, bacteria are everywhere. So unless you literally sterilized your water, yeah, you got bacteria in there. That's just the way it goes. All right, that brings us to tube 1. Tube 1 has, first off, color change, which lets me know some acid has been produced and the brilliant green has changed color. But a change of color is not the pass. What's my pass? Oh, that's right, gas. Gas is my pass. So here we go. I have got gas here. I have now, what do I know about tube 1? I've got a gram negative because I have survival. And not only is a gram negative, it is a lactose fermenter with gas production. Oh, that's awesome. This is my pass. I mean, it's not awesome if it's your well water. Now you're really concerned. You know, for most people, their wells at this point have failed. And usually the only people who are moving forward are people who took water from, like, you know, a fountain or a creek or something like that, but okay, it could have been somebody's well. All right, we go forward. We go forward with this tube, and so what would happen is that this person that has the gas in their tube would be having to loan their tube to a bunch of people because most people are going to fail. All right, so we would go forward with the tube with the gas. And now we would take 1 sterile loop full of that positive gas, okay, so positive confirmed test, positive brilliant green lactose bile broth, the positive is the gas. take one sterile luteful and we would just streak it out onto eosine methylene blue agar, EMB. Now, we just did mannitol salt a minute ago, and it might look from these images that they're the same. They're not. This EMB is actually more purpley, and mannitol salt is more orange pink. So I'm sorry if they seem like they're the same. If you saw them side by side, you would not confuse them. So eosine methylene blue has two dyes, eosine and methylene blue. Eosine is red, so you've heard of eosinophils and stuff. So eosine is red, methylene blue is blue. Together they make kind of a purpley red. It's more red than purple, but still. Both of those dyes inhibit gram-positives from growing, so I still have a selective situation going on here. Okay. And if there's E. coli in here, I will get a really distinctive result. This is one of my favorite labs. But this one I really like. Eosine methylene blue is taking advantage of the fact that E. coli produces a ton of acids when it ferments compared to other gram negatives. So what's in eosine methylene blue agar? You don't need to know the specific composition, like the percentages, because that is just too hard to keep that in your head. But there is peptone in here. Peptone, okay, remember that's proteins. There's the agar, that's indigestible. There's now 2 fermentable sugars, not just lactose, but also sucrose. So if they can ferment sugars, they've got lots of sugars to work with, which will give me lots of acids on the other end. And then I have 2 different dyes, the eosine, which is red, the methylene blue, which is blue. So the 2 sugars provide lots of fermentation opportunities. Okay. And moving forward, we remember that E. coli, if we looked at this slide, that E. coli would actually produce a lot of acids. So it would produce lactic acid, succinic acid, and acetic acid, which is like vinegar, right? So we've got 3 acids produced here versus enterobacters, which are very common. They're in soil, which is, you know, soil's everywhere. It could easily be in your well. It's definitely in a creek, right? Okay. So enterobacter makes ethanol just like E. coli does. Ethanol, that's not acidic. Lactic acid, okay, same lactic acid. Formic acid, okay, formic acid. Butanediol, acetone, these are not acidic. Those are not acidic end products. Carbon dioxide is not acidic, and neither is hydrogen gas. So ethanol E. coli makes way more acids than other gram-negative bacteria. So E. coli is going to produce a ton of acids. EMB agar is designed to find E. coli. And so remember that E. coli is the test organism because it is 1% of the bacteria found in human feces. So that's what we're really concerned about. So we're going to be the ability of E. coli on EMB. And E. coli will give you these distinctive metallic green colonies. It's incredible. You cannot mistake this. These are the best tests when you just look and it's like, yes, there it is. Not like you're rooting to get E. coli, okay, in your sample, but it's not one of these tests where you're like right on the cusp. I don't know. Is it a yes? Is it a no? This is so obvious. You will get a metallic green colony for E. coli and E. coli only. And the reason is all those acids that E. coli makes, lactic acid, succinic acid, acetic acid, react with the eosine and the methylene blue to give you this distinctive color, this metallic green. Enterobacter orogenes is a gram-negative bacilli which also ferments lactose and does make lactic acid, but it doesn't make as many acids as E. coli. It will grow on this surface of this agar, no problem. That's not going to be a problem for it, but it's going to look sort of like purpley. Sometimes people say sort of a mucusy color. It's not this, okay? It is not the metallic green. So when you get that, then I come around and tell you that you have dirt in your well or dirt in your water sample, and that's typical. Hey, dirt's everywhere. So once again, I'm looking at EMB here, which has two special dyes that react specifically with E. coli. Remember that EMB agar has two sets of sugars, lactose and sucrose. And remember, sucrose is a disaccharide. So is lactose. There's actually, really technically 4 monosaccharides in there, right? And so there's a lot of fermentable ability there. And so E. coli will give you a lot of acids from that, giving you this metallic green. Other organisms don't. And the typical other organism that you might find is Enterobacter aerogenes. Now, Enterobacter aerogenes is one of the 8 organisms that I would have given you in the unknown. And this reaction on EMB is something to take note of, because you might get as your unknown EA, Enterobacter aerogenes, which would have passed all the way through the coliform analysis, but here, oops, it's not E. coli, right? It's different, but it is a result that we can work with and we can remember.